Hello everyone and welcome back to my channel Be Yourself. This is Dr. Nisharma and today I'm going to deal with the second part of the explanation of part C questions from CSIr-JRF NET of Life Sciences, which was held on 18th of June 2017. In my first part I already have explained the questions up to 100. Now I will explain the rest of the 45 questions of part C. Let's start with the first question of this video. The first question is, researchers wanted to study light reaction during photosynthesis by blocking photosynthetic electron flow during the herbicide dichlorophenyl dimethylurea and paraquat. The researcher listed the following observation, and we have to find out the incorrect statement from the fourth statement. The first statement is that before reading this statement, I must say you that DCMU basically block the pigment system 2 and paraquat pigment system 1. So start reading these options. Both DCMU and paraquat block the electron flow of pigment system 2. No. Both DCMU and paraquat block the electron flow in pigment system 1. No. Rather, it will block DCMU will block pigment system 2, whereas paraquat will block pigment system 1. So the answer, both of these are wrong statement. Here, sorry, DCMU blocks electron flow in pigment system 1. No, it will block the pigment system of 2. So this statement is also wrong. The only statement which is correct is that DCMU blocks electron flow in pigment system 2 while paraquat blocks in pigment system 1. So the incorrect answer will be A, B and C. Next one. Following are a few statements regarding water potential in plants. Solute concentrations and pressure potential contribute to the water potential of the plant cell is in a given state. It is true because we know that water potential is equals to the solute potential and the pressure potential. We represent it somewhat like this. So the statement is right. Next is, when a flaccid cell is placed in a solution that has a water potential less negative than the intracellular water potential, water will move from the solution into the cell. It is true because if we are saying that water potential is less negative, it means what? It is having a higher value. We know that negative means the one which is having the less than 0. So one which having the less negative, it means will have the higher value, and water potential move from the less negative to the more negative part, or we can say from higher potential to the lower potential. So the statement is true. Next is, when a flaccid cell is placed in a solution, it has a water potential less negative. It means more potential than the intracellular water potential. Water will move out of the cells into the solutions. No, water potential of the solution is higher, so it should move from the solution toward the cell. So the statement is wrong. Water potential of a plant cells under severe water stress is always less negative. If water potential of the plant which is having a severe water stress, and it will have the less negative, or we can say the more potential, then what will happen? Water will start moving from the cell toward the solution. But if it will happen, then plant will undergo permanent plasmolysis. So it should not be there, rather it should be more negative value, so that water, it can take water from the solution and the cell may gain some of the gain some of the water for its survival. So statement is again wrong. So the statement which is correct is only A and B. Next one, quite simple questions. The following scheme shows the flowering status of the plant species and photoperiodisms in which it is grown. L denotes the light period and D denotes the dark period. Now we can say that the in only two diagram has the flower plant, other is not supporting the flowering, and in both of two, light period should be the limitations for the flowering, and the second is that the period which is determining the flowering status is the dark period. So the species is a short day plant. From here we can say that it is a short day plant, and length of the dark phase determine the flowering status. So the option will be number 1. Next question. In a photo response experiment, embedded seeds were kept under the following light regimes, and their germination status was denoted as follows. So, in complete darkness, there was no germinations, and uh, red along with the darkness, red light along with the darkness leads to the germination. Then again, red light, far red light, and again this darkness which is not germinating. So we can see the germination in the two places. One is here and one is here, and in the both the cases, sorry, not germination, germination here and here, and in the both the cases, dark phase is followed by the red phase. And we know that for the seed germinations we need the PF. So it means that in the presence of red light, P will be converted into PF. So the statement should be correct is that the red light is converting P into PF, and PF promotes the red seed germinations. In next also, if you will see here, the P is converted into PF, but again with the presence of far red light, PF has again converted into P of Phytochrome. So if it will proceed with the darkness, then it will not germinate, rather it is requiring the PF for its germination. Next question. Following are the few statements regarding the structure of terpenes. Before going further, I would like to say something about the terpenes. Terpenes is actually the monomer unit of isoprene, which is having a 5 carbon compound. So one having the 2 isoprene units, or we can say that in total 10 carbon terpenes is known as monoterpenes. Example is geranyl diphosphate. One having a 3 unit of C5 is known, or having the 15 carbon terpenes is known as sesquiterpenes, or farnesyl diphosphate is the example. 20 carbon terpenes is known as diterpenes, and geranyl diphosphate is the example. The larger terpenes, which include the diterpenes, have the 30 carbons, tetra having the 40 carbons, and polyterpenes generally have the N number or more than 8, or we can say more than 40 carbon compounds. Now move to the question. First says isopentyl diphosphate and farnesyl diphosphate are monoterpenes and sesquiterpenes respectively. Isopentyl is a 5 carbon compound, so it is not the monoterpenes, because the monoterpenes has the 10 carbon, so the statement is wrong. Next is squalene. Squalene is a 30 carbon compound, and geranyl diphosphate are triterpenes and monoterpenes respectively. The statement is true. Next is dimethyl allyl diphosphate and geranyl diphosphate are having the 10 and 20 carbon respectively. No, it is not so. Diterpenes have the 20 carbon compound. It's true, whereas the sesquiterpenes have 15 carbon. So the statement is true. So the answer will be number 2, which is B and D. Next question is, consider the following facts regarding the control of shoot apical meristem size in Arabidopsis. So these are some statements from where we have to analyze the pathway which is controlling the SAM size. Now first statement is loss of CLP1 gene leads to the bigger SAM. Means that if uh, CLV1 gene is not there, it will lead to the bigger size of the SAM. It means it is controlling negatively the size of the SAM. Next statement is loss of CLV3 genes leads to the, again it is the negative regulator of the SAM size. Third statement, loss of WUS gene leads to the small SAM size. It means WUS is positively regulating the SAM size. Fourth statement, when CLB1 and WS leads to the small size, it means CLB1 has some negative regulation to WUS also, because of this a small size of SAM1 is producing. Next statement, when loss of both CLV3 and WUS leads to the small size, it means again it is also controlling the WUS size, WS productions, and it is indirectly controlling the SAM size, decreasing the size of the SAM. So it is not, from this statement, it is not clear whether it is directly controlling the WS or it is or this is having some connection between these two. So move to the next statement. Loss of both CLV1 and CLV3 leads to the bigger SAM. So even if both will be not there, then WS will be present there, and it will switch on and it will regulate the bigger size of the SAM. Next statement is overproduction of CLV3 leads to smaller SAM. It means when CLV3 is overproducing, then it is negatively regulating the WS, and because of which the normal size, the bigger size is not there. Rather, it decreases the size of SAM and leads to the smaller SAM size. Next is overproduction of CLV3 in the loss of function mutant of CLV1 leads to bigger SAM. It means it is controlling indirectly to the WS with the help of CLV1, because if it is overproducing, then it is leading to the mutations or a loss in the function of CLV1. So when CLV1 will be lost there, then it will not able to control the WS, and the WS will able to control the bigger size of the SAM. But if it will be produced in less amount, when CLV3 will be in the optimum amount, then CLV1 will be expressed and it will halt the process of WS and leads to the smaller size of the SAM. So if it is overproducing, it is controlling the CLV1, which because of which the production has been stopped. It is negatively regulating the CLV1, and indirectly it is has switched on the WS, because it is not, because of absence of CLV1, the functional site of CLV1, WS will be switched on and it will lead to the bigger size of SAM. So as per this, the correct pathway should be number 3. Next question. In kidney, sodium ion is reabsorbed across the second half of proximal tubule due to the positive transepithelial voltage and by other mechanisms. The following proposed statements could explain the development of this positive transepithelial voltage is. Actually, transepithelial voltage is positive there because of the diffusion of chlorine ion from the proximal tubules. So the only statement true is here is number A. So the answer will be number 1. Next question. The action potential of the nerve fiber is described by different components, including after hyperpolarization. The mechanism of generations of this after hyperpolarization has been proposed in the following statement. We know that the affinity for the potassium ion is more than in comparison to the sodium ion. So whatever the hyperpolarization will be there, that will be only due to the because of the potassium ion. So the statement here, let me read it out. The increased conduction of sodium ion has returned to the baseline level, but the conductance of potassium ion remain elevated. It is obviously be there because the affinity for potassium ion is more. Next statement is the membrane potential is pulled even closer to potassium ion equilibrium potential at the after hyperpolarization phase. Obviously it will be there. And the two statement here, the conductance of sodium ion is increased. No, the conductance of potassium ion will increase, not the sodium ion. After the after polarization phase, the membrane potential is driven closer. No, it is just analogous to that of the B. So the answer, which is both correct, is option A and B. Next statement. In the experiment of healthy young men, the muscarinic receptor antagonistic atropine was administered to one group A, while the beta adrenergic receptor antagonistic propanol was administered to another group B, in 4 increasing doses of equal concentrations of both drugs. The effect of two drugs on heartbeat are shown below. So we know that the atropine will block. Atropine will leads to, or it will affect the parasympathetic, and beta adrenergic receptor will affect the sympathetic response. So the statement we have to find out the incorrect statement. So incorrect statement is atropine and paratropine blocks the sympathetic. No, it will block the parasympathetic, whereas propanol will block the sympathetic one. So it is just opposite. As the change in the heart rate is more in the group A than in group B, the sympathetic tone is usually predominant in healthy resting individual. No, it predominates in the parasympathetic, rather predominates in the healthy resting individual. So the statement wrong is here A and B, while the statement C and D, which is atropine and paratropine, block the parasympathetic and sympathetic effect on the heart respectively, is true. As subsequent changes occur in the heart rate with atropine, the parasympathetic tone is predominant in the healthy resting individual. So statement right is here C and D, but we have to point out the incorrect statement. So the answer will be A and B. Next question. The partial pressure or value of hemoglobin for oxygen is increasing during exercise. The mechanism of where these changes is described in the following proposed statement. Increased carbon dioxide productions by muscles elevate the partial pressure of carbon dioxide of blood, which affect the partial pressure value. Obviously, we know that partial pressure controls the exchange in the hemoglobin. So the statement here that the increase, if partial pressure will be more in the muscles, then it will exchange, oxygen will be delivered to the muscles, and from here carbon dioxide will be exchanged to the RBCs or with the hemoglobins. So answer is right. The affinity of hemoglobin for oxygen increases as 2,3. No, the affinity of oxygen decreases in presence of biphosphate glycerate. Another is increased body temperature shift the hemoglobin dissociation curve to the left. No, it will dissociate the dissociation curve toward the right. So the statement is wrong here. And here we have to point out the incorrect statement. So the incorrect statement is B and C, because D, the decrease in pH of the blood reduces the affinity of hemoglobin for oxygen, is true. Because in the first, in my video, I think in the same year, in 2017 June, in I think that was a question was in there in buffer of blood was there in section B. So in there I have explained how the carbon dioxide value will increase and decrease the pH of blood, and that will leads to the increase and decrease to the pH, and it will affect the binding affinity of oxygen there. So I'm not going to explain it again. Next question. There is evidence that following prosthetic stimuli, cytokines produced by the CNS causes fever, possibly by local release of the prostaglandins. Accordingly, the following statements has been proposed, and here we have to find out the correct statement. So the first statement is that cytokines act independently and directly on thermoregulatory centers. It's true. The intrahypothalamic injection of prostaglandin receptors and agonistic will prevent fever. No. Statement is wrong. Antipyretic effect of asparagine is exerted on the hypothalamus to prevent the prostaglandin synthesis. It's true. And aspirin blocks the infections and eventually prevent fever. No, aspirin doesn't block the infection, rather it regulates with the help of hypothalamus gland. So the statement true is here A and C. So option 4 is the right one. Next is following are some statements about the mechanism of stimulation of receptors for touch, pain, vision, warmth that may or may not be correct. We know that touch receptor is controlled by the help of subchannels. It can be sodium potassium channel, sodium calcium channel. In Chewimi plant, or you have seen that the touch receptors is controlling the osmosis there with the help of sodium channel. So the touch receptor actually is required the voltage gated cation channels for its activation. So the first statement, the touch receptor does not require any voltage gated channel, is the wrong statement. Next is that light causes closing of sodium channels in the outer segments of the rods and cones. It's true. Pen sensation is caused by opening of sodium ion or sodium calcium ion in the free sensory nerves. Hence, it's true. The one receptor is activated by non-selective anion channels. It is false, as it is very specific for the ion channels. So the statement true is here B and C, and we have to point out the correct one. So the answer will be the B and C. Next question. The following is the schematic diagram representation of the hypothetical pathway involved in the formation of eye color in an insect species. So one which having the white eye pigments X and Y both will control the white eyes color of the of the insect. So and here X is converted into the vermilion, or the white pigmented is converted into vermilion pigment with the help of gene A, and gene B will convert the white pigment into brown pigment. And when both the pigment gene will be there, it leads to the mixing of the pigment and leads to the red eye color. Next statement, it is stating there A and B genes are the linked. So there will be no crossing over between A and B. And again it is stating that the female with genotype A positive A and B positive B are test crossed. Further, in these females, the two genes are linked in Cs. So A plus and B plus represents the wild type alleles, while the A and B represents the null alleles. So if it is saying that it is in Cs form, then it will be like this: A plus and B plus will be on the same chromosome, and it is homologous chromosome. It will have the A and B, and it is linked and having the distance of 10 centimorgan. So we have to test cross the A positive A and B positive B. So what will be the chromosomes here? It will be A positive B positive and AB chromosome. When it will form the gamete, it will not undergo independent adsorbment as per the Mendelian law, because it is the linked one. So it will produce only two gametes as B positive, A positive, as A positive, B positive, and AB. And it is a test cross, so it will test cross with the recessive one, which is, and it will give A positive, B positive, and AB, and again it will give AB and AB. So one is giving the wild type and one having the both the genes, it is giving the red one. But here we are having that the expected ratio, we have to find out the expected ratio for all the four one. And next point here, it is given that it is 10 centimeter in distance. So 10 centimorgan, sorry, I'm telling centimeter. So here we have the red eye and white eye ratio is the 1:1. One will have the red eye and one will have the wild eye, and in between this, which is forming due to the presence of A and B genes. So as the distance is 10 centimeters, so we will directly subtract it 10 minus 1, or the 9 centimorgan for the A and B respectively. So the ratio should be 9:1:1:9. So the option will be number 3. Next question. Yes, this is based on the Cre-lox hypothesis, or sorry, Cre-loxP recombinase value. In this, we have to find out in the following diagram, segment A and C are the copies of 10 base pair repeats DNA sequence flanking a unique stretch shown by B, and A and C is in inverted orientations is as indicated by the arrow. So Cre is the protein which is directly responsive for the knockout experimental basis, and Lox is the one whose orientation will decide, and it can undergo leads to the translocation, or it leads to the inversion, or it can also lead to the complete deletion. So if it will be in the inverted positions in both the sides, then it will lead to the deletion of this flanked region, which is B. Inversion of this flanked region. So the option correct here is that only B will be inverted. In my upcoming videos, I will explain you in detail about the Cre-lox recombinase activity, because if I will explain here, it will become it will take much more time, and I will not able to upload that much large video in YouTube. So I am better reducing my content here. In my upcoming videos, I will deal with this topic, Cre-loxP recombinase activity. Next question is very simple, interesting question is there. Somatic cell hybridization is used to assign a gene to a particular chromosome. When two cell lines from two different species are fused, they form a heterokaryon, which tends to loss of the chromosomes as they divide preferentially from one species. A plane of the cell lines was created from the mouse and monkey somatic cells function. Each line was examined for the presence of monkey chromosomes and for the production of a given enzyme. So here it is given the plus is showing that enzyme is produced here, negative is showing that enzyme is not there, and the presence of monkey chromosomes 1 to 10 chromosomes is there, and the plus is showing that the chromosome is present when the cell line A is producing the enzymes. And now here we have to find out the chromosome which is basically responsible for the enzyme production. So very simple trick here is that first point out the chromosomes which is not producing the enzymes. So here if minus is there in C and E cell lines, enzymes is not produced, but even chromosome 2 is there and chromosome 8 and chromosome 9 is there. If this chromosome will be responsible for the production of the enzyme in cell line C, then it should be absent there. But if it is present there, then it is obvious that the role of chromosome 2, 8 and 9 is not in the production of enzyme is not there. Next in E, cell line E, we have the chromosome number 4, chromosome number 5 and chromosome number 10 is there. So obviously these chromosomes is also not playing any role in the presence of enzymes. Now come to the positive one. Positive one is A, B, D and F. So in A, enzyme is there. If chromosome 1 will be responsible for the production of the enzyme, then it should be present in all A, B, D and F, but it is absent in case of B. So it is also not the one which is responsible for the production of enzyme. Move to the chromosome number 3. It is present in A and B, but it is absent in F, but here the enzyme is producing. So again it is not responsible. Move to the chromosome number 6. It is absent in A, but enzyme is produced there, so it is also not be the answer. The option only present here is here 7, as it is present in cell line A, B, D and F, and this is the only chromosomes which is responsible for the presence of enzyme. So the answer will be number 2. Next question, a phenotypically normal fruit fly was crossed to another fly whose phenotype order was not recorded. Of the progeny, 3/8 were wild type. Wild type means one having the E positive, VZ positive and VZ positive. And wild type and 3/8 had ebony color. 1/8 ebony color means which is the recessive one is here ebony color. 1/8 is had the vestigial wings. It means VZ, and 1/8 is having the ebony color and vestigial wings. So what should be the other parent? If the one parent is phenotypically normal, it means, and it is also producing the recessive one, so it will be heterozygous in all its for its allele. So it will be VZ. So now here the option is that it is producing the wild type, and wild types means the one gene which is always expressed in the populations. So if it is the other parent will be A positive for the one of the site, then it should be heterozygous because it is also producing the homozygous recessive one. So it will be E positive, E, and for vestigial wings also it is producing the wild type having both having the positive value and both having the negative one. So it will be also in the heterozygous form. So the option here will be number 4. In normal individual, there are 3 MS2 restriction sites, 2 flanking the beta globin genes and 1 within the gene. In individual affected by a gene, a single nucleotide polymorphism in the beta globin gene abolishes the internal MST2 recognition site. RFLP pattern for this locus obtained by hybridization using a probe internal to the flanking MST2 site from three siblings of a family as shown here. So if it is having the one having the normal one will give the two bands for as it will flank the MST2 recognition site is there in the B globulin. So when it will cut the beta globulin gene, it will give the two band. So here it is clear also here that it is a normal daughter cells. But the one it is normal son is there, but it is also having a one which is the affected or the one which is the carrier one. But in the affected son it is the recessive one. As if it would be dominant one, then it will not be normal one, rather it will be the affected one. And it is autosomal recessive because if it is present in the sun. So what we know that sun will have this chromosome X and Y chromosome, and X will express the gene. So if gene will be present there, then this is not possible for a sun to be normal. So the answer will be autosomal recessive. Next question. In a transduction experiment using A positive, B positive, C positive genotype as a donor and A negative, B negative, C negative as the recipient, A positive transductants were selected and screened for B and C. The data obtained are below. Here we have to find out the co-transduction frequencies of A positive and B positive and B positive and C positive. It means if it is A positive, B positive and C positive, then co-transduction means the one in F minus we should have the A positive and B positive alleles. So genotypes. So to find out the A positive, B positive, we have to from the data we have to check out where A positive, B positive, how many recombinants are there. So A positive, B positive recombinants here is 98 and other is 168. 98 plus 68, and if you will sum up the total, you will get 750. Now similarly, we will take for the B positive and C positive one. So if you will take the C positive and B positive, C positive, here we have only one which is having B positive and C positive. So for the B positive and C positive, it should be 68. So what we do to find out the frequencies, we add the 68 plus 68 by total number, which is 750. It will be for A and B transductions, positive, B positive, and it will be for B positive, C positive transductions, 68 by 750. When you will calculate it, you will get approximately 22%, and in this you will get approximately 9%. So the answer will be number 2. Next question, I'm not going to deal with these questions. I'm directly coming to the answer because it is totally based on the definitions. No need to explain here. So answer is number 4. Next question. Given below are some of the statements on vertebrates. Which one of the following statement is incorrect? First statement is muscular post-anal trait and pharyngeal slits are derived character in vertebrates like the notochord and dorsal hollow nerve cords. These are not the derived character, rather these are the shared characters. So the statement is wrong here. Next is here. So we have to find out only one statement wrong. So I am not going to read further. The only statement wrong is here number 1. Next question also I'm not going to deal with this. I will directly tell you the answer because this is also totally based on your memory. And based on certain definitions, match the following types. The answer is number 4, which is A, B, and E. Next question again, it's the disease and causing or causative agent. The answer is here. It's again number 4. Answer is here. Again, match the following. The answer is number white blood. It's number 3. You better match it out and mug it out. Next question. The following is a list of reproductive structures found in vascular and non-vascular plants. Which one of the following combination represents the structures primarily associated with the gametophytic life cycle of the plant? So the gametophytic life cycles of the plant is including the archegonia and megaspore, and the one is the pollen grain. So the answer will be number 2. Yes, this is all based on the formula. The net reproductive rate R naught is 1.5 for a given populations. If NT of the population is 500, so we have to find out the number of populations after n plus 4. N plus 4 means if we have the net reproductive rate is R naught, so after 4 years, what we'll have? We will have 10 total number of population will be the initial populations into the net reproductive rate. So the answer will be 500 into 1.5 into 1.5 into 1.5 into 1.5, and it will give you number 2, which is 52531.250. Yes, this question is from the Lotka-Volterra model. So question is quite simple. If once you know the what exactly the model want to tell, so I will not explain it much, but in my I will make a separate video on Lotka-Volterra model. I will just explain the what on related to the points related to the questions. The question is that two species M and N occupy the same habitat. Given below is the space graph in which the abundance of the species M is plotted on the x-axis and the abundance of species N is plotted on the y-axis. For each species, the zero growth inclination is plotted. So this zero growth inclination is this line, and this is for the N species, and the solid line is for the M species. So the concept is that when a point is within this dotted line for the N species, then the N species will increase in size, or the species will increase in the population, and above the line there will be decrease in the number of N species. Similarly, for M species also, the area inside anywhere inside this line there will be increase in the species of M, and above this line there will decrease in the number of M species. So if you will point, we have to point out the incorrect statement. So here it is saying at a point B. So at a point B, what will be there? As it is the point below the solid line, which is for M species, and above the dotted line, which is for the N species. So as it is above the N species, so the N species will decrease at this area, but the M species will increase. So but here it is written that at point B, population of the species N is increasing, rather it is decreasing there. So the statement wrong, incorrect is number 3. Next next question is, which one of the following statement is not correct? The statement here is that the herbivore enhance the productivity of the productive ecosystem. Is true. The terrestrial based food chain is longer. Obviously, it will be longer, and the consumption efficiency of herbivore is higher in grassland than ocean. The statement incorrect is the number 3. As the consumption efficiency of herbivore in ocean is more than that of the grassland. Again, this question is totally based on the definitions, what the ketone species is, what the indicator species is, and so I'm directly telling you the answer. You better match with that of the options, and just read out what exactly the ketone species means and what the exactly indicator species means. So I'm not going to explain it. Next question is again based on the data. As per the National Air Quantity Standard for the India, which of the following option gives the correct concentration limits of various gaseous air pollutants for the resident area? So the answer is the one is as per the National Quantity Air, the limit is the numbers SO2 and NO2 40, O3 is 100, and O is 02. Next one, the plant is visited by the bats during the night and sunburn during the day. Given these informations, which of the following character best match this plant? The plant which is visited by sunburn is the tubular and red in color. And the plant which is visited by a bat can also feed on the tubular nectar plant, because the bat having a long tongue, so it can directly lick the nectar from the tubular plant. So the answer is the plant is a shrub with the tubular red diurnal flowers. Next one is the western honeybees collect nectar and pollen from the flower. The following a few hypotheses proposed to explain this behavior in Apis mellifera is actually this hypothesis is in the past those individuals that fed on nectar and pollen left more descendants than those who prefer only nectar or only pollens. And another hypothesis is the ancestor of the honeybee was dependent on some sugar and protein rich diet, and the honeybee have inherited the same taste perceptions. So the answer will be number 3, which is A and D. Next question is again here based on the match the following and totally based on the we have to just work out the answer. So the answer here is Jean-Baptiste. I am directly telling you the answer. So the answer will be here 2. Following diagram represents various ways in which a character may evolve. So these are the 3 diagrams which is showing how the characters are evolved, and here dark dot is representing the derived traits and circle is representing the ancestor one. So move to the first diagram. Here it is saying that the one of the ancestor is having the derived traits and it is passing it to its descendants. Such types of transmission is known as synapomorphy. A is synapomorphy. Next is number B. Here it is shared by the 2 sets of this species, but it is not present in any of its ancestors. So such type of character is known as homoplasy. So option B should be homoplasy, and the one which is derived in the only one species and very unique in the tree, it is having very distinctive feature, is known as autapomorphy. So the answer will be number 4. Next question is, in circadian rhythms studies, following may be the possible generations for the effectiveness of the light entrainment to the day and night cycle. So what happened for the cardiac rhythms? We generally prefer for the bright light, and the second is that dim bright light along with the dim light can basically affect the relatively more. If we are using the bright light instead of dim light, then in comparison to dim light, bright light will have the greater response. But if we are using the dim light in comparison to the darkness, then the response for the dim light is more for that of in comparison to the darkness. So statement correct here is only B and D. Next question, which of the following statements regarding the endosymbiotic hypothesis of the origin of eukaryotes is incorrect? The only incorrect statement here is that the event of engulfment of photosynthetic cyanobacteria by host cells was primitive to engulfment of alpha proteobacterium. It is not primitive, rather cyanobacteria is more primitive in comparison to the alpha proteobacterium. So the incorrect statement is number 2. Next is, to understand the singing behavior of the bird, the following 3 characters were measured. First is the territorial character represented by the graph A. B is the female fertility, dotted line, and song rate is for sharp dotted line, which is representing the C. So now from here what we can analyze, when the singing behavior is increasing with that of the territory, it is also aligning the territory. Another is that the egg laying period, which is also been increasing with that of its singing behavior, and also the fertility. So what does it state? That male birds sing as a display of strength to rivals and to attract females. Next statement is also match the following types. So the answer will be number 3. Next question is given below are the 4 statements regarding the genetic transformations of plants in the laboratories. So here we have to find out the correct statement. So for the genetic transformations in the plant using the agrobacterium, so what should be the correct statement? The plant incapable of sexual reproduction cannot be transformed by agrobacterium tumefaciens. It is not true because we are not using the agrobacterium tumefaciens for the sexual reproductions. Rather we are infecting the media having the plant tissue culture. So the statement is wrong here. The other statement which is correct is integration of the transgenes in the organelle genome occur primarily by homologous recombination. Actually, it's true. It is basically take place in chloroplast and it flanked the new genes which is to be integrated in the desired plant. So statement is true here. Next statement is the enhancer trap construct used in agrobacterium media transformation could contain a functional coding sequence of a reporter genes and minimum report and a minimal promoter. Actually, it is the characteristics while we are designing the one for the for the infection with the agrobacterium, that it should have the functional coding sequence as a reporter genes and the minimal reporter promoter should be there. So the statement which is correct is here B and C. So the answer will be number 2. Agrobacterium-mediated transformation was used to generate transgenic plants using a construct with the selection marker gene X and a passenger gene Y. Expression level of Y protein is 8 independent transgenic plant are given below, but it is showing various variations. So what can be the possibility for the variations? In the previous questions also it is indicating that the reporter genes and minimum promoter should be there, which is the characteristics of agrobacterium. So now it is somewhat linked here. Here is that the position effect of passenger gene. So it will be affected by the positions where we are inserting the genes, whether how far it is from the promoter and how it is located. And this is particularly known as position effect. So this can be the one of the reason. And another is that the more the copy number will be of the passenger gene, the more will the chances for the integrations into the plant cells. So the answer which is correct here is A and C. So the option will be number 1. Quite simple questions. If a breeding experiment, two homozygous parental lines P1 and P2 were crossed to produce F1 hybrids. Due to an experimental error, seeds of these hybrids don't got mixed up with the seeds of the two other germplasm lines P3 and P4, and the hybrid seeds derived from them. A marker-based fingerprinting exercise was performed using 6 random selected seeds. F1 to F6 are there. This hybrid seeds. So from the mixture of the material, we have to find out using their bands pattern. So it is quite simple to analyze this. The answer is number 3. How? If you will look here, that P1 having the band, it is present in F1, and the number second band is also present in F1. Next band is also present in F1, and move to the P2. P2 has also F1 has also band that of the P2, and again band of P2 is present in F1, and again it has the band for the P2. So the cross of P1 and P2 will give the F1 progeny, and if you match the other one, it will not get the similar bands in other progeny with that of the following options. So that's the answer is number 3. NMR spectrum of the 30 residue peptides were recorded at 25 degrees Celsius. The following observations were made. The first observation is that the NH and C-alpha H resonance were well resolved. This is because of the hydrogen bonding in the NH. The nose spectra showed extensive NI and Ni plus 1 conjugatives, because this is the special characteristics of the alpha helix, and it is more extensive in case of alpha helix when it will compared with that of the beta helix. Again, the NH resonance shows the slow exchange with the deuterium. It is so because H will be exchanged with that of the deuterium. And the answer is here helical conformation. I have also listed some of the characteristics of the alpha helix when we will examine it under the NMR. So these are the certain characteristics, and here in the options there were the two characteristics it is matching here. One is the 1 plus I plus I plus I plus 1 consecutive frequencies, and another is the exchange of hydrogen with that of the deuterium exchange. So I better pause the video and note out these points for the alpha helix. I will make a separate video on the NMR, and then I will show you how to analyze the data for the secondary structure of the proteins in NMR. Next question is again match the following, so I will not going to explain it. It is based on the biostats. So I am telling you the answer. The answer is number 3. Next question. In an experiment designed to clone a PCR amplified fragment in a cloning vector designed with XO1 and SMA1, which one of the following combination of restriction enzyme can be used in the PCR primer to generate the compatible int for cloning? For the compatible int for the primer designing, we must have a hanging end. So here and the only it is giving it will give the hanging end, but it will give the blunt end. So first we will target this hanging end one. So when we will come, we'll show this. The SAL1 will give the hanging ends, which is TCGAC, TCGAC, which is the complementary to that of our desired one. And regarding the blunt end, we have to use some adapter or linker to make it hanging to design a primer against it. So the most appropriate one is here is number 3, which is the SAL1 and ECO R5. Next question. The researchers was working with 3 proteins A, B and C, which may have the potential roles in gene expression. In order to validate the hypothesis, electrophoretic mobility C was performed. The purified proteins were allowed to bind with the labeled DNA, and the results obtained after the auto-radiography is shown below. As it is saying that protein A possesses the DNA binding motif, it is true because if you will see here, the protein A is having the DNA binding motif. Next statement is protein B possesses DNA binding motifs. DNA B is not having the band, so it is the statement is not right here. Next statement is protein B binds to DNA protein A complex. So when A and B is both present here, then we are getting the 2 bands, one with that of the A and other is for the B. Next statement is protein C binds to DNA only when protein A is bound. So here when B and C is present, we are getting no bond here, but when A is present, we are getting one band only, and that is true also of that of the A. So the statement is wrong here. We are not getting the band for the C. So the only correct statement is A1 and 3, and the option will be number 2. Last question is protein group is symmetric operations such as inversion and mirror plan are not applicable to protein crystal. This is because it will have the equimolar concentrations of DRN form, and this is known as handedness. So the protein molecules will become handedness, and because of this reason it is not applicable. So this all were in these videos, and my upcoming videos I will discuss about the Lotka-Volterra models and the Cre-loxP recombinations system, which is basically used for the knockout studies. So don't forget to subscribe my channel, be yourselves, and click the bell just straightforward to the subscribe button so that you can get the notification as soon as I will upload my videos. And very soon I will also upload videos on the questions